Hello everyone, good evening. Please let me know if you are connected. Are you guys in here or should I wait for you? Alright, so we'll move on to another technique right now. And before that, if anyone new is joining, I think I've already introduced myself. I'm Saniya Pamnani and this is the Unacademy profile. You will find that in the description below. If you have not yet downloaded the Unacademy app, you can download the Unacademy Learning app from Play Store. You can use my code SPLive10, which will give you access to all the free resources. There are many free classes that I've taken even on YouTube. This channel, Let's Crack IIT JAM, please refer to the playlist in case you need help with metabolism, genetic crosses, or biochemistry. I have taught all of it. And these are the plus courses, guys. So, in case you are planning to subscribe to Plus, you can definitely get these recordings. You will get daily live classes as well. You will get live tests and quizzes to evaluate your preparation. You will get structured courses and you will get unlimited access. So, therefore, you can even cover up your preparation for physics, chemistry, and maths. These are the subscription costs. Once you apply the code SPLive10 in the code section, you will get a 10% off. Also, you can use credits. So, if you've been watching my lessons and if you have taken special classes, you will be getting some credits which you can use for further discount. If there is any content that you feel is inappropriate, you can definitely report it and you can claim your price. So, in this series, 10 days, 10 techniques, we have finished quite a lot. I will just summarize what all we have done. So, in case you have missed it, you can definitely go back and watch these lessons. So, in the first day, we did Agarose Gel Electrophoresis. On the second day, we did SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis. On the third day, we covered Western blotting. In the fourth session, which happened today at 4, we covered DNA cloning. And today I am going to teach you PCR in this session. Alright, so let's get started. Just give me a minute, I will switch to the presentation. Here. So first of all, we are going to do types of PCR and we are going to understand what PCR is. I have taken a couple of classes already on PCR, so I'm assuming that you will have a basic understanding of what polymerase chain reaction is. However, if you do not have that, please do not worry, we will go through it. I cannot see anyone live. I don't know why. Okay, I am waiting for you guys to come live. Alright, shall we begin? Okay, so PCR is nothing but your polymerase chain reaction. If you have something that you need to amplify, so what we do is we use a efficient and a cost-effective molecular tool which basically amplifies or copies your small segments of your DNA or your RNA. PCR combines your principle of complementary nucleic acid hybridization with those of your nucleic acid replication that are applied repeatedly through numerous cycles. So, what this means is that once you have a small segment of DNA that you want to clone or you want to, amplify and you need it in a large quantity, the principle that is applied behind using a PCR is nothing but your complementary nucleic acid hybridization, that means your ATGC sequences, and your nucleic acid replication. So the same way how your replication happens, right? The same mechanism is used here as well. And guys, if you are not aware of you rules of replication or you do not know the basics of replication, please join my special class tomorrow. It is absolutely free. There's no charge that I'll be imposing. So it's absolutely free. You can come and attend that lecture. Yes, Prerna, hi, good evening. I didn't see you in the previous class today. We did an important topic, DNA cloning. So please go and revise in case you didn't find time to attend it live. Where are others? So yeah, nucleic acid replication. If you guys do not know the rules of replication, please attend the special class tomorrow. And just a reminder, today at 8:30 p.m. also we have a class about Ramachandran plot. Prerna nucleic the class for replication is also at 8:30 p.m. tomorrow. Okay, I'll be giving you the links in the Telegram channel. So in case you have not yet joined the Telegram channel of Let's Crack IIT Jam, please do so. All right, so let's move ahead. Your PCR is nothing but a polymerase chain reaction which uses the principle of your complementary nucleic acid hybridization, that means A base pairs with T and G base pairs with C, with those of nucleic acid replication that are applied repeatedly through numerous cycles. So please remember that this process of amplification, it happens in uh, numerous cycles. Okay, it's not just done in one goal, unlike your other techniques. So it results in an exponential production of a specific DNA sequence by a factor of 10 raised to the power 7 within a relatively short period. So do you have an idea what that short period is? So that short period is not minutes, but it's in hours. So it takes about 2.5 to 3 hours to run a PCR. All right. So these are the components that we require. Why do we okay, no. Let me reframe my question. What is that machine that is used for PCR called? Any idea? What is another name for the PCR machine? Quickly. What do you have any idea? What is it called? Okay, I think no response. That I'm assuming then nobody knows. It's called a thermocycler. Okay, why do we call it a thermocycler is because it does nothing but change temperature. Okay, so there's a slot wherein we will put our PCR tubes, and there will be settings that you have to adjust which will tell the machine that okay, for first 30 seconds this should be the temperature, for first 1 minute this should be the temperature, then for next 30 minutes this should be the temperature, for last 3 minutes this should be the temperature. So for every step the temperature needs to change, and that machine is not doing anything but only changing the temperature. So what do you do is you take a PCR tube, you put all these components, you load it into the PCR machine, and that's all. Your work is done there. It's very simple. You just have to take out the components and then analyze. We'll learn how. First of all, let's understand what are the things that we will put in your PCR machine. Okay, so first thing is your DNA template. DNA template is the sample that contains the target, okay, target sequences to amplify. You will have DNTPs, which are deoxyribonucleoside triphosphates. You will have a PCR buffer. You will have primers, reverse and forward, and you will have Taq polymerase. Now, two important components here are primers and Taq polymerase. Primer is nothing but a small sequence of, see, if this is the segment of your DNA that you need to amplify, what happens is, first of all, this will be separated, okay, split into two. Then you will have a primer which will come and attach here. And like I said, this works on the rules of replication. So in your replication, you should remember that your DNA polymerase is an enzyme that cannot start anything from scratch. It always needs a primer. And that is why we need both a forward primer and a reverse primer for this replication or for this amplification to follow. And then you will have your DNA polymerase or your Taq polymerase, which is similar to DNA polymerase. It's actually a DNA polymerase that is obtained by Thermus aquaticus, which is used and that does your polymerizing process. Okay, so moving ahead to the steps. Okay, so first step that is going to happen is denaturation or your stand separation. Basically, in this step, what happens is that there will be separation of your two hydrogen bonded complementary chains of DNA into a pair of single-stranded nucleotide molecules by a process of heating. So when we put the sample, when we have the sample in the PCR tubes, we will just expose it to a high temperature like which will be around 94 to 96 degrees Celsius, and that will help in the separation of the two strands. Why do we need to separate two strands? Is because we need to amplify it. It needs to undergo replication, and for that, you know that strand separation is necessary. Next will be annealing. The temperature here is lowered to 45 to 60 degrees Celsius so that your primers can attach themselves to the single-stranded DNA strands. So depending on what kind of DNA you want to amplify, first of all, you have to design primers. Primer designing requires you to know the base sequences of your gene of interest or the DNA, the short DNA strand that you're trying to amplify. If you know the sequence of that, then you can, form your own primers. You just have to submit a request, and there are several pharmaceutical companies also which provide uh, ready-made primers. So you don't have to do that step on your own. All you have to do is add the required dilution of primer inside the solution or your Eppendorf. Okay, not Eppendorf, PCR tubes. Actually, PCR tubes and Eppendorfs look the same, but PCR tubes are like much smaller in size. And for that, you need to lower the temperature so that they can go and bind to the single-stranded DNA. Then there is extension, that means synthesis of your new DNA. It starts at your annealed primer and works its way along your DNA strand, and the temperature required is 72 degrees Celsius. So extension means once you have your DNA, you have your primer attached to it. Now it will pick the your Taq polymerase will pick the nucleotides or your DNTPs that are there in the solution, and it will start joining. Okay, so this strand will start getting extended. Okay, now one thing that you need to know here is that this is Taq polymerase. So why we use the Taq polymerase is because it can withstand high temperatures. Withstand high temperatures because it is derived from a because it is derived from microorganisms that grow in very warm environments, not even warm, in hot springs actually. Okay, so they can bear this temperature of 94 degrees Celsius to 96 degrees Celsius, and that is why we are kind of employing Taq polymerase. In case we use human DNA polymerase, what will happen is during the denaturation step. See, please remember you have everything inside your PCR tube, right? It's not, if the temperature is changing, everything inside this will have to withstand that change, right? Will have to undergo that change. So, if you have a human DNA polymerase instead of a Taq polymerase, that particular enzyme here will get degraded and denatured it, and then it won't work, right? So therefore we needed a special kind of DNA polymerase that will be used for your PCR. And please remember, with all three steps, see here you have 94 to 96 degrees Celsius, here you have 45 to 60 degrees Celsius, and here you have 72 degrees Celsius. So the only change here, like the major change here, is temperature, right? So that thermocycler that you will use will do nothing but change temperature depending on the requirement. So it will do the denaturation step for, let's say, 1 minute, it will do the annealing for, let's say, 3 minutes, then it will do the extension, let's say, for 36 minutes. So 30 minutes or something. So depending on your gene of interest, depending on how long your primer is, and depending on how many cycles of extension you want, this will undergo temperature changes, and then you have to set cycles of this. Okay, so let's say I want 30 cycles, or let's say I want 40 cycles or 20 cycles, depending on what I require. Okay, now once you have taken out the samples, once the PCR is over, you use Agarose Gel Electrophoresis to view. Okay, so what you'll do is you'll just load your sample with a loading die. You'll mix your sample with a loading die. You'll load it onto your gels. You'll run the gel, and you will see, corresponding to the ladder, if your amplification has happened and if it has come to the correct size. Okay. I hope this is clear to you. Now there are different types of PCR that are present. One is your real-time PCR, reverse transcriptase PCR, multiplex PCR, nested, high fidelity, fast, hot star, GC rich, long range, arbitrary. But all of these PCRs, they run on the same principle. Okay, just that the use and the application is different depending on what we are required to do. So what you can do for homework is figure out what is the differences between these PCR. Okay, and I think you have understood already what basic PCR technique means. So these are few applications: identification and characterization of your infectious agents, direct detection of microorganisms in your patient specimens, identification of microorganisms grown in the culture, detection of antimicrobial resistance, investigation of stainless relatedness of a pathogen of interest, genetic fingerprinting. So in your genetic fingerprinting, what happens is sometimes you have a sample, but the sample is not, huge enough, or it's not sufficient to do DNA fingerprinting, right? So what happens is we do PCR and we amplify the product, and then we can use it for paternity testing so that we do not run out of sample. Detection of mutation, investigation of genetic diseases, cloning genes, and PCR sequencing. So there are several things wherein PCR are used, and plus when you figure out what are the differences between these techniques, you will be able to appreciate that PCR, though it's a simple technique, though not much is required for us to do, but we will, but it has a very broad application base. Okay. So here is a diagrammatic representation. You can see that you have parental strands. They will melt at 95 degrees Celsius, 94 to 96, whatever. then they will anneal to your forward and to your backward primer. The next step will be extension. So your forward primer will attach here. So this direction here will always be 3 dash. Okay, so this will be 5 dash, this will be 3 dash, this will be 5 dash. Here also this will be 3 dash, this will be 5 dash, this will be 3 dash, and this will be 5 dash. Okay, yeah, so this will be 3 dash. So that's because your polymerase is only capable of adding nucleotides to the 3' end. The free OH here on your 3' end is going to attack the incoming DNTP, and the phosphate bond breaks up, and the DNMP is attached, right? Is attached to your DNA sequence, and that's how your amplification happens. So it will extend. Now, in the second cycle, also, what will happen is this entire DNA, this here, will separate, right? They will denature. So you have a red strand here, you have a black strand here, you have a black strand here, you have a red strand here. Now, now you had two strand, and in the next cycle you will have four. So similarly there will be different cycles that we will be running. Okay, there will be, let's say, 30 cycles will be running. So that depending on how much amount of amplification you want to get, that many cycles you can run, and you can have your DNA in whatever is the required quantity. Okay, is it clear to all of you? Is it clear to all of you? So that's all about your PCR. If you want to subscribe to Plus, you can use my code SPLive10. Also, there is a offer going on. If you take 6 months of subscription, you get 1 month free. If you get 12 months of subscription, you get 2 months free. Okay. So let me know if you want to buy your subscriptions, and please, what to say, subscribe to the channel, Let's Crack IIT JAM, so that you get further updates. Also, you will need to join the Telegram channel in case you're not very active on the Unacademy platform. All the special classes information will be given there. Okay, and yeah, I think that's all for the class today. So yes, I'll see you tomorrow in another session, which will be at 5 p.m. with another technique. Also, if there are any topics that you want me to cover in special classes or YouTube platform, then you can let me know. I will see if I can take your suggestions. And if you like the channel, please hit the like button. If you feel that this was helpful for you, do share it with your friends and subscribe to Let's